the transition state and they lowers the activation energy. Okay. So generally, where the rate of the reaction is right, the rate of the reaction is inversely proportional to the activation energy. If the rate of the reaction is more, the activation energy is less, and the activation energy less, the rate of reaction. Okay, so this is the inversely proportional. So generally, when these particular enzymes are involved in a chemical reaction, so they will speed up the chemical reaction and they will decrease the time and they increase the rate of the reaction here, right? So the time is inversely proportional to the particular rate of the reaction, and they will decrease the activation energy. So after the reaction, enzymes release back. So means the properties of this catalyst. Generally, they will involve in the chemical reaction, and finally they will get their original state, same as that catalyst property. The enzymes also they will involve in the chemical reaction, and they will get back their structure or the geometry, whatever the changes are occurred. Finally, they will gain again. They will gain the original state of that enzyme. For example, here you see this carbonic anhydrase enzyme. Carbonic anhydrase enzyme. It's catalyzes the hydration of the 6 carbon dioxide molecule per second, because the enzymatic reactions are very speedy process. They are very quick processes here. So you see, this particular carbonic anhydrase enzyme, they will catalyze the hydration of the carbon dioxide, right? So how many carbon dioxide molecules will get hydrolyzed with this particular carbonic anhydrase enzyme here? So 6 here. So you can assume, right? So you can assume that how much means how many molecules are converting into the product here. So that is the speed of the reaction here, which is done by this particular enzyme. Here you see this the diagram with free energy, that is a G, okay, delta G, and a rate reaction coordination. So you see this is here is a so this is the normal reaction, right? So this is the normal reaction where it is having this much delta G, right? So delta G activation energy, this much energy is required. Uh, this much energy is required here. It's the uncatalyzed reaction, the normal reaction here, without enzymes, without catalyst here. And when the same reaction is carried out, the presence of a enzyme, with the enzyme catalyzed reaction, you can see the decrease of the rate of the reaction. So this much difference is there, right? You can see the difference of this. So this much difference is there. So that is a specialty, that is the importance of your enzymes. Speed up the reaction and it's uh, they will speed up the reaction and they will decrease the activation energy. So here the whole enzyme molecule cannot interact with substrate here. So a part of that enzyme. Okay, so generally the enzyme, the chemical reaction, the whole enzyme will not be involved in the chemical reaction. They won't interact completely. A part of that enzyme will be interacts with molecules in the reaction or the molecule substrate here. So every enzyme molecule have a specific groove or the hollow or a deep irregular three-dimensional cavity. That is called the active site. Okay, so every enzyme is having its own active site, and that active site is the point for the substrate molecule. So, for example, here you see, okay, so that reaction is that particular reactor site, is, that cavity is called the active site. And this active site has generally all the active sites are having chiral linkage. Chiral the end groups of this functional group of amino acid at inside is helping the so binding to this specific substrate, the transformation of the functional group of the substrate. Okay, so the enzyme binds to the substrate and its active site, hydrogen bonding, water wall uh, interactions, ionic interaction, dipolar interactions, hydrophobic interactions. So this particular enzyme, the substrate molecule can be interacts and binds with your enzyme active site with various means, various interactions, and that interaction you can see that hydrogen bonding, the ionic interaction, dipole interactions, and weak water molecule interactions. So all these are the interactions which is able to bind with your receptor active site. So substrate molecule can be forms a bond or the interaction. So that the interaction it may be the weak bond or it may be the strong. Then in general, you can see that the interactions like here you see the hydrogen body weak interaction, ionic interactions, and water one interaction. All these are weak interactions. Only the covalent interaction is a strong interaction. Yeah. So, but generally, it is less preferable. The covalent interactions are less preferable. Only we can observe all these kind of the ionic interactions, hydrophobic interactions, dipole interactions, hydrophobic interactions. Here is a, you can see the diagram where the active site of an enzyme, where this the irregular cavity, the irregular geometry of that particular enzyme is called the active site. So this is the right. So this is the site where that particular substrate molecule or the drug molecule is going to interact or bind with the site. For example, you see that pyruvic acid reduced to yes, lactic acid, the presence of the alcohol, because there's enzymatic reaction where your pyruvic acid is reduced to the yes, lactic acid. So in the presence of the alcohol dehydrogenase. So how this enzymatic reaction takes place, you can see here. There is pyruvic acid. So here is that pyruvic acid where is undergoing enzymatic reactions in the presence of lactate dehydrogenase, right? Lactate dehydrogenase, the enzyme which is converting this pyruvic acid to lactic acid. Right? So it is undergoing it. So when it is undergoing that reduction reaction, okay, lactate dehydrogenase, the presence of NADH and So it's action, spike acid, uh, so and six stages. How the changes, how this stages, the active site of your engine, active dehydrates. So if the active engine, okay, and you see this, right? So this is your pyruvic acid, and this is all these are the cofactors, and there is a residue, summary acid. Is composed with the active site is composed with various acid residues and coenzymes and cofactors. So in this particular lactate dehydrogenase enzyme active site is facilitating for the occupying or the inserting of your pyruvic acid, and this pyruvic acid is forming the various interactions, various body interactions with the carbon acid. See here, right? So this pyruvic acid is, this is forming the ionic interaction here, ionic interaction. And here is a hydrogen bonding interaction, hydrogen bonding here, and here is a, right. So there you see this the methyl, right. So there is no hydrogen bonding donor groups, and there's no hydrogen bonding acceptor group. So it is hydrophobic. Okay. So hydrophobic interactions. So in this way, you can see the various possible interactions. Okay. So it will be the pyruvic acid will be occupying the receptor active site. Receptor nothing but your lactate dehydrogenase is forming the various interactions. Then in this active sites where the reaction is takes that substrate molecule is converting in the product where the NADH, okay, so it is giving the proton, right? It is transferring this proton here to this carbonyl group, right? And it is converting into alcohol. Let's see this conversion of this CO group is we have the NAD, right? So it is transferring here, then this is coming, it is converting to alcohol. So that is a reduction stage, basically. Right? Next, mechanism of enzyme catalysis. So mechanism of enzyme catalysis. So here we know that enzymes are specific in nature. Enzymes are very specific in nature here. So yeah, the specific nature and the catalytic activity of enzymes can be explained by the following models. Right. So these are the models which can be explained the enzyme catalysis property and its specificity property here. Catalytic activity and specificity activity can be explained by the following models. Like you can see this lock and key model here, right? So you get the lock and key model, industrial fit hypothesis, and the 3-point contact model, right? So these are the various types of the mechanisms which can be explained the enzyme catalysis. First, we'll see this lock and key model here. So as per this lock and key model, so generally this lock and key model is, okay, yeah, the lock and key model is proposed by Emily Fisher, right? Emily Fisher is one of the famous chemists, scientist here, chemistry scientist or chemist here. He was proposed this lock and key model. And as per this lock and key model, where the enzyme acts as a lock and the substrate acts as a key here. So the substrate molecule must have the correct shape or the geometry shape. Nothing. So this model assumes that the enzyme molecule has a rigid structure and it is having a fixed shape and its active site. Right. So this is very first model proposed by the Emily Fisher. And as per this model, right? So where the substrate molecule must have the the substrate molecule must have the matched geometry or the correct structure with the receptor active site here. Right? The mismatching of the geometry or the shape will not be fit into this particular receptor active site here. Okay, this is the proposal of this lock and key model. And so here this model, right, proposed because where this at that time they assumed that this particular receptor active site, right, so where the enzyme molecule is a rigid structure and it is having a fixed shape, right? They thought that it is a having fixed shape and it is rigid structure. That's why they proposed that the substrate molecule must have the exact or the correct shape or the geometry which is fit into the your enzyme active site here. Okay, that is your lock and key model. And this model indicate that substrate specificity of the enzyme. But the disadvantage of this model is it is not because the enzyme is not rigid molecule and it can be changes its conformation. In generally, the enzyme is not a rigid molecule model here, where it can be change its conformation or the shape here. But they assume that the enzyme is a rigid structure. That's why they propose that the substrate molecule must have the correct geometry or the fixed geometry shape with your receptor active site. Right. So then one more model is one more hypothesis is proposed that is called the induced fit hypothesis. So induced fit hypothesis is it is proposed by the Coastland. It's proposed by the Coastland scientist in the 1968. And according to this model, an enzyme molecule has a flexible structure. Okay, so you can observe the difference of these two hypotheses. With the lock and key model or the lock and key hypothesis, where it is proposed that the particular enzyme's geometry or the enzyme structure is fixed or it is rigid structure. But in the induced fit hypothesis, it is proposed that okay, so where the enzyme molecule is having the flexible structure here. So when the suitable substrate molecule is approaches the enzyme molecule, right? The enzyme molecule, when that particular suitable substrate molecule is approaching your enzyme molecule, then it induces a conformation changes in the enzyme molecule. Okay. So when a suitable other respective substrate molecule is approaching your enzyme molecule here, then whatever the geometry is there for the enzyme molecule here, so that enzyme molecule will be makes changes its geometry according to the geometry of your receptor. According to your substrate molecule here. Okay, so that is called the induced fit mechanism. Means where your substrate molecule brings the changes in the conformation, right? Conformation of the receptor active site, and it will be binds or it will be sit in that particular receptor active site. Okay. So that a proper alignment between an active site of enzyme molecule and the substrate molecule is established. Okay. So due to changing the conformation of your receptor active site according to your substrate molecule, so you can see a proper alignment or the proper assignment or the uh, insertion of that your substrate with your receptor active site is takes place, and you can see a good interaction is established here. So this model explains the reaction specificity of this enzyme, how the enzyme can interact with the different substrate which have the undergone same type of reactions. And this model also having some disadvantages where this the disadvantage of this model is that so it cannot explain the enhancer selectivity of enzyme. Okay, enhancer selectivity of enzyme. So enhancer, right? So enhancer selectivity nothing but where the stereoisomer, right? So where the diastereomer enhancer, right? So where the selectivity when that the spatial arrangement of the atoms, right? When the change in the spatial arrangement of the atom is takes place, then whether it is binding is takes place or feasible or not, so that point is not explained in this particular inducible mechanism. And one more model is proposed to explain the enhancer selectivity is the three-point contact model. Three-point contact model here. So three-point contact model where the enzymes are an enhancer selective and they are capable of recognizing and selectively binding to the particular enantiomer. So as per this model where that enzymes are so they will recognize that enantiomer and it will be bind with that particular enantiomer. Where the R and S, for example, the enantiomers are having one is R configuration, another is having S configuration. So one will be binds with that one, another will not have the same kind of interaction. So then it will be recognized, it will be differentiated also. The enantiomers can be differentiated with this particular three-point contact model. So the enantiomer discriminating ability of the enzyme active site is explained by a three-point contact model, which is and this model is proposed by your Izon and Steadman. Okay, Eson and Steadman, these two scientists proposed this three-point contact model. And according to this model, an enzyme active site is have at least three contact points here. One in among these three contacts, which two are used for the binding to immobilizing that substrate, and the third contact point is stereochemically depending and forms the basis for this discriminating the enantiomer. That means one enantiomer gets all three interactions or the contact points correctly, while other is not having got correctly here. Because you see where the okay, the orientation of the groups, orientation of the atoms will be different. So generally, if one enantiomer is having three linking the binding interactions, another will not have that same kind of interaction because the orientation is different from that one. So therefore, only one enantiomer can enter into the active site and it can bind with that particular active site, and the other it is totally excluded from the active site there. So this model explains the enzymatic resolution of the racemates, and this model is valid only for the chiral molecule generally. Here is you can see here the S isomer and R isomer both are there. So where this S isomer and R isomer are interacting with your chymotrypsin, right? Digestive enzyme where the chymotrypsin, right? And it is converting into your the products here, the amino acid and some other derivative is there. So here the S isomer only fits and it have three contact points with the active site of your chymotrypsin enzyme. And the R isomer does not have any means this kind of three interactions here. One it have the different, like it may two interaction or one interaction. But S isomer is having three contact interactions here. And so it will be bind with that receptor active site and it converts that, it gives some products like amino acid and some particular uh, okay acid, right, acetate here. There is you can see the diagram where this here is amino acid 1, AA1, AA2, AA3. Here, three amino acids. Residues are there in the receptor active site. Okay, and we are the particular S isomer. Okay, the compound S isomer, which is having these three kinds of interactions, where one is a hydrogen bonding interaction, and the ionic interactions and the hydrophobic interactions. Okay, you can see the right here is a hydrophobic interactions. And here is ionic interaction here. And here is your that hydrogen bonding. Hydrogen bonding. So in this way, we have the various interactions where the S isomer. You can see these three interactions can possible only for the IS isomer, and it cannot possible for your R isomer, right? It does not forms any means this kind of interactions. So you can discriminate, you can differentiate both the isomers with your enzyme active site here. And you can say compare with these interactions S isomer and R isomer. Here you can see the R isomer having only two interactions where it is having the hydrophobic interactions and ionic interaction, and where the hydrogen bonding interaction is not present in the R isomers. Okay? Next, holoenzyme, cofactor, and coenzymes. So these are the some terminologies where. Okay, so generally all the enzymes are protein in nature here because the proteins modified into enzymes, and some enzymes are simple protein molecule and some are the conjugated proteins which are associated with the non-protein part in the enzymatic activity. And generally, these enzymes are called the haloenzyme here. And this haloenzyme consists of a protein part called the apoenzyme, and other part is called the cofactors. Right. So in a enzyme, we have the two parts where the right apoenzyme and the cofactors. And where these cofactors are the small organic molecules or the metallic ions which are required of these ions here. And these are the activators you see. It's zinc carbonic carboxy peptidase, carbonic anhydrase, right? Where this iron is present in the heme. Zinc is present in the carboxy peptidase and carbonic anhydrase here. and copper activator. Other cofactor is present in the peroxidase enzyme here. Peroxidase enzyme, sorry, peroxidase enzyme. The coenzyme, where these are the organic compounds which its molecular weight is around 400 and it's weakly bind to the active site here. These are required in stoichiometric amount for the catalytic activity of your enzyme. And these coenzymes are transient carriers of small ions or the group in the substrate. Yes. Here the classification, the nomenclature of the enzymes, and this is very, very important. Find where the how to classify the enzymes and how to nomenclate these enzymes. Right. So here the classification and nomenclature of your enzymes. So generally, the enzymes nomenclature, right? The enzyme nomenclature is very specific and it is very unique here, where the all the enzymes name is end with the ase. Okay, so in the name, any compound name or the molecule name is end with the ase suffix, then it indicates the enzyme here, right? For example, here you see the maltase dehydrogenase here. Okay, lactate dehydrogenase, maltase here, amylase. Okay, all these are right. So where it is having the ase sound in the end here. Okay, and there are several enzymes which are not specific for the substrate. Therefore, here in 1972, the Commission of Enzymes, EC, okay, Commission of Enzyme, uh, is one of the International Union of Biochemistry, or IUB. Right? It is one of the Commission of Enzyme. It is one of the International Union of Biochemistry. It is established in 1972, and this was proposed some systematic nomenclature and the classification of these enzymes. So here we see the classification of enzymes. Where here is the enzymes have been classified into 6 groups here, 6 categories. You can see that categories where the oxidoreductases, transferases, hydrolases, liases, isomerases, ligases. So these are the 6 groups of the 6 categories of your enzymes. We'll see about this more about your oxidoreductases, where these enzymes catalyze the biochemical oxidation and reduction reactions. The name itself is telling that the oxidoreductases. Okay, oxidation as well as your reduction. They will involve in the biological oxidation and reduction reactions. And generally, these are energy yielding reactions here. These are the energy gaining reactions. And these further classified into there are six types of oxidoreductases here. In that we have that dehydrogenase enzyme, where they catalyze the removal of water of 2 hydrogen atoms here. Right, so it can be remove the water of the 2 hydrogen atoms is the dehydrogenase enzyme, and oxidases. So this catalyze the addition of oxygen and oxygenase. So it is catalyze the incorporation of oxygen into the substrate molecule, and oxidative deaminase, where they catalyze the oxidation of amino acids, oxidation of amino compounds with the formation of ammonia. And the hydrolases, so the function of this hydrolases is, so they will introduce the OH groups in the substrate molecule here. And peroxidases, where they use the peroxide, hydrogen peroxide as oxidant. So these are the functions and functions of various types of categories of your oxidoreductases enzymes here. Here is the example. You can see that here the lactate enzyme, it is undergoing that dehydrogenase in the presence of lactate dehydrogenase enzyme. Okay, so you can see here the removal of two hydrogens from this lactate and it is converting into pyruvate. Next, transferase enzyme. So generally, these enzymes which catalyze the transfer of chemical group like alkyl, methyl, aryl, carboxyl, formyl, glycosyl, phosphate groups here, right? So they will catalyze of transfer of the groups here. The name itself, transferases, means they will transfer the groups. So what are the groups can be able to transfer? Yes, methyl, ethyl, isopropyl, carboxyl, acyl, formyl, glycerol, phosphate. So these groups can be transferred from one group to another group, one substrate to the another substrate here. Here you see A plus B plus CD. So you can see the AC plus BD. So the exchange of the the exchange of the groups are that atoms is takes place here. Next, hydrolysis, where you can see this include the hydrolytic enzyme which catalyze the hydrolysis of carbon CO bond here, CC bond and CN bond. Okay, by addition of water. So when you add a water molecule where the CO bond or the CC bond or CN bonds gets hydrolyzed here, here is you see A plus AB plus water. When the water is added to this AB molecule, where the water is it is reacting with A and it is expelling your B. Okay, so this is the hydrolysis and the esterases. This catalyze the hydrolysis of carboxylic esters. It's the ester where it is converting into alcohol in the presence of lipase enzyme here. Ester is converting into glycerol. Next, peptidases, where the hydrolyze the formation of peptide bonds in the proteins. Next, liases. You can see this lysis. Generally, the lysis is a cleavage. Okay. So they will catalyze the removal of specific groups like water, carbon dioxide, ammonia. And generally, these are the reversible reactions. In this, we have the example is decarboxylase in decarboxylases, where it catalyzes the addition of the removal of carbon dioxide. There is a aspartate where upon decarboxylation, in the presence of aspartate decarboxylase enzyme, so it is going to form your this particular the amino acid here. Next, isomerases. Isomerases means they catalyze the interconversion of optical or the geometrical or the positional isomers, where the isomers gets isomerized, right? The isomers where you can see what are the isomers here, where they're having the same molecular formula but the different properties is called the isomers here. So these particular isomerases, they will catalyze the interconversion. The interconversion of isomers like optical isomers, geometrical isomers, and structural isomers here, positional isomers. And in this, we have four categories like cis trans isomerization, racemases, epimerases, and interconvertases. Okay, here is the isomerase where you see this isopentenine pyrophosphates. Okay, so where the bond exchange of this, right? The shifting of the bond from one carbon to another carbon. Only the bond shifting is taking place. Rest all groups are, all the atoms are same in this particular, in both cases here. The A and B both compounds here, where it is equilibrium state reactions. Here is isopentenyl pyrophosphate is converting into, right? Here is isopentenyl pyrophosphate, IPP. IPPE. It is converting into dimethyl allyl pyrophosphate here, DMPP. Okay, IPP converts into DMPP. Next, ligases. So here is a ligases are the synthases. So these enzymes catalyze the joining together of two molecules which coupled with the breakdown of phosphate bonds in the ATP. Now you see the nomenclature of enzymes. Generally here the International Union of Biochemistry, IUB, it has given the nomenclature for this enzyme in 1960. Okay, it is given the nomenclature for this enzyme in 1960. And each enzyme, it has assigned a systematic code number here. And this enzyme commission EC with the 4-digit number. And where the first digit indicates the major class of the enzyme. The second digit indicates the subclass here. The third digit indicates a subclass. The fourth digit is specific or the substrate here. So these are the enzyme commission is proposed. Enzyme commission is assigned in nomenclature for this enzyme. And this particular enzyme is each and every enzyme is having its specific code, and each code is having indicating some names here. Where, for example, here the EC is the enzyme commission 11127. Okay, EC 1116, EC 2711. So where the you can see this lactate dehydrogenase, right? Like glycerol dehydrogenase. So all these are the some kinds of the subclass and subclass here. Okay, and the factors substrate one. Right. So now we are going to this factors affecting the enzyme catalysis. So generally here the enzyme catalysis will be affected with some factors. Those factors are the enzyme, right? The structure of the enzyme here. Then the substrate molecule here, and the physical agents like pH, temperature, and redox potential. All these are the factors which affecting your enzyme catalysis and effect of enzyme concentration. First, we see the effect of enzyme concentration. Where this the enzyme concentration, right? So with increase of enzyme concentration, the rate of the reaction will be increases because if the enzymes are more available, then more substrate will be converted into product. So where the availability of the enzyme or when the enzyme concentration is more, then the rate of the reaction will be more here. It is what is that? Directly proportional. Next, effect of substrate concentration. So by keeping the enzyme concentration constant, so with increase of substrate concentration, with the rapid increase of rate of the reaction is takes place initially, then when the concentration continues, it is where the rate of the reaction will be decreases because where the availability of the substrate is less, so no conversion of the product here, right? So initially it will be more, and later, okay. So because we are kept the enzyme concentration constant here, we are kept the enzyme concentration constant, but we are providing more substrate here. So though the substrate molecules are available, but the enzymes are not sufficient to convert that substrate into products here. So the initially you can see the tremendous increase of the rate of the reaction at the optimum point here, at the optimum points where the decrease of the rate of the reaction is takes place. And the effect of temperature. So generally, okay, so most of the reactions, right? Many of the reactions cannot see any catalytic activity. Most of the enzymatic reactions will not carried out at 0 degrees temperature here, because at 0 degrees, the kinetic energy of the molecule is less or it is negligible, right? So with increase of the temperature, the enzyme activity or the rate of the reaction will be more, and that increase will be maximum to 40 degrees Celsius. Okay. So with increase of the temperature from 0 to 40 degrees, the rate of the reaction will be increases, and beyond that 40 degrees here, beyond that 40 degrees, the rate of reaction will be decreased, because you see the enzymes can bear the temperature up to 40 degrees here. So beyond the 40 degrees, the enzyme becomes denatured, right? The enzyme becomes denaturization. At a higher temperature, the enzyme cannot survive. The enzyme cannot exist here. So it becomes denaturization here, right? That's why the means the enzyme activity, the rate of the reaction will be maximum at 40 degrees temperature here. Below 40 degrees, up to 40 degrees. But beyond that, you can the where the decrease in the temperature means decrease in the rate of reaction is takes place here. Okay. So this is about the enzymes. So I'm stopping here.